Welcome to AP Biology. Today I want to talk to you about viruses. We're going to take a very quick tour through this. What you can see here is a bacterial cell, and it has a bunch of little viruses on it. These viruses that infect bacteria are called bacteriophages, and so we'll talk about those a little bit. We're going to start with virus structure in general. So viruses are not made out of cells. They don't have cytoplasm or like all the organelles and stuff that you would find inside, ribosomes, that kind of thing. So what is a virus? A virus is a small infectious particle, and it consists of nucleic acid. That nucleic acid could be either DNA or RNA, so we'll talk about that a little bit in a minute. And it's enclosed in a protein coat. So all viruses have genetic material, DNA or RNA, and they have a protein coat around them. Some of them also have a membranous envelope around that, which is made from the membrane of the host cell, but it's still not a cell because it doesn't have all the other stuff that you would find inside of a cell. So here are just some examples of what viruses might look like. All of them will have genetic material and a protein. Some of them, like this one, also have a membrane around the outside. So viral genomes can consist of either double-stranded or single-stranded DNA and either double-stranded or single-stranded RNA. So that's different from cellular life. We have DNA as our genetic material, as do all life forms on the planet, as long as you don't call viruses life forms. So viruses can be DNA viruses, and this one's double-stranded. So AP might give you a problem where they say that 20% of the genome is adenine, and 20% is thymine, and 30% is 30% is cytosine, and 30% is guanine. And so you would have to know from here, first of all, that T-thymine, that means it's a DNA virus. And so if the genome of the virus has this, you would know it's a DNA virus, and then you would know that it's probably double-stranded if the numbers, the percentages of the A and T are the same, and the percentages of the C and the G are the same. If they give you a different sample, and they say there's A, T, C, G, the T will tell you that it's DNA. If it says that there's 20% A, but maybe 30% T, and I don't know, maybe 10% C, 30, what does that, 40 leave us? 30, 40, 50, 60, yeah, so 40% G. That's kind of a dumb number. But anyway, if the A and the T don't match up and the C and the G don't match up, then it's probably single-stranded DNA. RNA viruses, a virus genome can be made out of RNA. One type of RNA virus is a retrovirus, but there are other types, so we'll talk about that in a minute. If the sample is made out of RNA, it will say it has a UCG, so the U is for uracil. Again, if these match, like maybe, I don't know, maybe this is 30% and this is 30%, and then these would have to be 20%. Right? So if the A's and the U's match and the C's and the G's match, then it's double-stranded RNA. If there are A, U, C's, and G's, and the amounts of A and U don't, if the amount of A and U don't match and the amount of C and G don't match, then it's going to be single-stranded RNA. Okay, so the capsid is the protein shell that encloses the viral genome. All viruses have that. The bacteriophage, the virus that infects bacteria, probably the coolest looking ones, but there are plenty of others. The influenza virus, lots and lots of different viruses. Okay, some viruses also have viral envelopes. So these are membranous envelopes that come from the host cell. And so you can see them here. Here's the virus budding out of the cell, and the part right here that's enclosing the virus, that's a membrane, right? That came originally from the host cell. But not all viruses have those. Virus specificity, that's just the idea that a virus infects one particular species for the most part and doesn't infect other species. Now, that's not totally true. There's a range of hosts. So this just means that viruses, like one particular virus, doesn't infect every single organism out there. There's either one species or a small range of species that a virus will usually infect. So most viruses only infect specific host cells, like, for example, most viruses that infect humans don't infect cats, but there are some that can infect both. Each virus has a host range, so a limited number of host cells that it can infect. So general features of viral reproduction. First of all, the virus cannot replicate outside of the host cell. So the virus can replicate only inside of the host cell. And that's not true only of viruses. There are plenty of parasites that can only replicate inside you, and some parasites that are intracellular that can actually only replicate inside your cells. So how is a virus different? Well, a virus has lost or doesn't have most of the stuff it needs to replicate. Like, it doesn't have its own ribosomes. It's missing most enzymes. It has like the bare minimum, at least for many viruses. It has its genetic material, maybe an enzyme or two, and kinda that's what it's got. You know, the protein that gets broken down as soon as it gets in, it's really got its instructions and maybe an enzyme or two. Some viruses are a lot bigger, but there's no virus that has all the stuff that it needs in order to replicate, like the enzymes that it needs and the ribosomes that it needs, all that stuff. Okay, so what are the steps? The viral genome enters the host cell, and the cell begins to manufacture viral proteins. So here's the virus. It enters the host cell, which is this. The proteins are, they sort of come off, and you've got the viral, either DNA or RNA. In this case, it's DNA. So it's going to use, typically it's going to use the host DNA polymerase and all the other enzymes needed to copy more of its own DNA. And then it will use it'll use the enzymes, the RNA polymerase, to make messenger RNA. And then it will also use the ribosomes to make the proteins that it needs. So this is transcription and this is translation, and it uses the machinery present in the host cell, the host cell's enzymes and the host cell's ribosomes in order to do that. And then the viral parts, the DNA and the proteins, will assemble and bud off and make new viruses. So the virus makes use of the host enzymes, the ribosomes, the transfer RNAs, the amino acids, the ATP, all the stuff inside the host cell in order to reproduce. Viral nucleic acid molecules and proteins will spontaneously self-assemble, that's what they're doing here, into new viruses. Sometimes they'll kill the host cell at that point and break out of it, and sometimes they'll just bud out like this. By budding, I mean they form like a little extension here. Here's an example of a question you might see where you have one type of virus and another type of virus both infecting the same cell. And when that cell, so the virus, both viruses will direct the cell to produce more viral genetic material and more viral proteins. And also maybe they'll bud out and they'll use the membrane of the host cell here, having its own, like the glycoprotein, carbohydrate protein. This outer membrane here will have come from the host cell, but it will have surface markers, proteins, carbohydrates, or glycoprotein, combination of the two, that the virus has directed. So anyway, these are two separate viral particles. They both are infecting the same host cell. When the new viruses are packaged, it's possible that a virus can be produced that has some of this one's genetic material and some of this one's genetic material, producing a new strain of a virus that can infect humans. That's not good. So that is one way that a new viral strain can be produced. Another way that a new viral strain can be produced is just mutations in the virus over time. They reproduce fast and a lot, and they can mess up when they make, that when they replicate their genetic material. Any mutation is a change, and that can be a new strain. So these new strains can cause pandemics or global epidemics. We've heard of those now, huh? Okay, next idea, bacteriophages or phages. Just in general, these are, could also say phage, you might hear that. These are viruses that infect bacteria. So these are the best understood viruses because bacteria are easy to grow, and so scientists have studied these more extensively. What you can see here is the protein coat on the outside, and see how it's injecting its genetic material inside. It's probably its DNA. So it's the DNA that goes inside. You'll look at some videos later where scientists actually used this in order to figure out what the genetic material was. But anyway, so the protein tailpiece attaches to the phage. It has to have a particular connection. If the virus so let's say this is one type of bacterial cell here. Maybe this virus doesn't, here's the virus, right? Maybe the virus doesn't infect another type of cell a different cell because it doesn't have the right surface markers to attach to. So anyway, the protein tail piece attaches to attaches the phage to the host and injects the phage DNA, the virus DNA, inside. So here is the viral DNA being injected inside. Notice that the protein stays on the outside, but the genetic material, the DNA or RNA, get inside the host cell. So that was just a repeat animation, by the way, just happens once. So phages have two reproductive mechanisms, the lytic cycle and the lysogenic cycle. The lytic cycle is a smaller word and it's quicker. So the virus attaches to the receptors. The virus attaches to the receptors. It's a very specific attachment. If it can't attach, then it doesn't infect that one. The protein for bacteriophages stays outside. The DNA or RNA gets injected inside. And then it just goes to town. It goes right away to start making more of its DNA with the enzymes in the host cell. And then it will use that the host enzymes to make RNA and then the ribosomes to make proteins. Then they'll assemble and then they'll break the cell dies, and that is the lytic cycle. So first there's attachment, and then the virus injects its DNA inside the host cell. And then the poor host cell, this bacterial cell, is now producing more of the DNA and some proteins for the virus, and then the viruses assemble. They don't grow like cells do. They don't start like little and cute and get big. They just assemble, and then they release, and the cell dies. So the host cell dies, pretty much right away. Okay, so to lyse is to break, so that's where the term comes from. Virulent phage is one that always goes right to the lytic cycle. Virulence just means like how bad or how deadly it is. Quick other note here: restriction enzymes. Restriction enzymes are something that bacteria has evolved to kill viruses. So if a virus injects its DNA, but the restriction enzyme can destroy the viral DNA. Like let's say the viral DNA, this stuff right here. Let's say the code was A C C T T. Yeah, let's do that. And then the other side would look like, no, that was a bad example. I'm going to try this one more time. Let's say the virus DNA read AACC GGTT, and the other direction would be the same, AACC GGTT. Maybe this particular restriction enzyme recognizes a restriction site right between the C's, and it cuts like this. If you cut the virus DNA, then the virus DNA doesn't work anymore. It's kind of dead. And if the cell, the bacterial cell itself, if its chromosome, its DNA has that site, it would methylate it so it wouldn't get destroyed. So anyway, that's a nice way that bacteria have to protect themselves from some viruses. Scientists have figured out how to use restriction enzymes in the lab. We will talk about that in the biotechnology unit. Okay, the lysogenic cycle is similar in the beginning. The virus has to attach to receptors on the surface of the cell. There's that specific attachment, so the virus is very specific for this type of cell, maybe that particular species. It injects its DNA or maybe RNA into the host cell. Let's just go with DNA for right now. The DNA then inserts itself, it'll double-strand itself, and then insert itself into the bacteria's chromosome. So we've got the black part is the bacteria's chromosome. This is the virus's DNA, and it just can get it can just stay in the genome for lots and lots of binary fission, lots and lots of times for the bacteria to reproduce, and then there'll be some trigger where it'll come out, and then it'll do the whole crazy lytic thing and kill the cell. So let's go over the steps of that. So with the lysogenic cycle, the virus injects its genetic material, let's say it's DNA, and here it is right here. We call that a prophage because it's not really the viral DNA well, it is the viral DNA, but it's sort of sitting there for like, hey, maybe later I might do something horrible, but I'm just hanging out for now. And then the cell does binary fission and reproduces, and now look at this. The cell has copied the virus's DNA along with its own DNA, right? So the the phage genome just gets reproduced each time that the bacterial cell reproduces. And that integrated viral DNA is called the prophage. That's this or this. Every time the host cell divides, it copies the phage DNA. So here's the virus infecting the cell. Here's the virus DNA that's become part of the chromosome, and then it got reproduced. And here's where sorry, I missed a little step. Here's the prophage where the virus genome has inserted itself into the bacterial chromosome. Here's binary fission, and now you have two cells, right here and right here, that both have that viral DNA in them. And actually, this is in a bacterial cell, but this can happen in our cells too. Viruses can insert their DNA into our chromosomes as well. In that case, they're called latent viruses. But anyway, back to bacteria here, what can happen is that there can be some trigger to send this prophage that was sitting in the lysogenic cycle back into the lytic cycle and destroy the cell. Bad stuff. Okay, so temperate phages are viruses that can sort of go between the two cycles. Okay, replicative cycles of animal viruses. So is it DNA? Is the genome DNA or RNA? Is it single-stranded or double-stranded? So these are sort of two key variables to look at when we classify viruses and how they reproduce. Like the last stuff I showed you was in bacterial cells. Viruses can infect people too, or animals. Actually, viruses infect pretty much every living thing on the planet. But I want to talk about viruses that infect animal cells or human cells right now for a little bit. So here are just some examples. The adenovirus, papillomavirus, herpes virus, pox virus. These are all double-stranded DNA viruses. Do not memorize it, but do know that some viruses have a genome that's double-stranded DNA. A's, T's, C's, and G's. Ooh, that is a really bad double-stranded DNA. That's better. Okay, single-stranded DNA, the parovirus. I guess it causes a mild rash. Anyway, it's got A's, T's, C's, and G's, but it's only one strand. And then there are some double-stranded RNA viruses like the Riovirus. Oh, rotavirus, I know that one. That can make you very, very sick, can kill babies, because they lose so much water with diarrhea. Anyway, double-stranded RNA. You may not have known that RNA can be double-stranded, but it can. The A's would match up with the U's, and the C's would match up with the G's. Okay, and then there are some single-stranded RNA viruses where the RNA serves as messenger RNA, where that right away does the RNA to protein thing. So, here's a notable one, coronavirus. I've taught this a whole bunch of times and never really looked too much at that. I'm like, I don't know what that one does. Sure know what it does now. And then there are some other single-stranded RNA viruses where the RNA will serve to produce another RNA, and this is the one that will serve as the messenger RNA, sort of like this is the template to make the RNA. Anyway, and then there's these single-stranded RNAs where the RNA serves as a template to make DNA, and then the DNA makes RNA makes protein. Those are called retroviruses, and those actually show up on AP tests like fairly frequently. So you should just know that viruses can have genetic material, genomes that are either DNA or RNA, and they can either be double-stranded or single-stranded, whether they're DNA viruses or RNA viruses. And then also, you should know a little bit about retroviruses. They are not the only type of RNA virus, but they're one, and they're really important because HIV is a retrovirus. And they're found in some tumors and stuff too. Okay, so a retrovirus is an RNA virus. And so what it does, here's HIV, I think, yeah. And so it's got a couple of strands of RNA, and so it also has an enzyme called reverse transcriptase, ACE. So if you remember the whole DNA makes RNA, are you sick of it yet? Makes protein. You might know, maybe, you might know that DNA to RNA is called transcription. And you might know that RNA to protein is called translation. So reverse transcription is the opposite of this. So let's look at that. Let's say you have some RNA and you use an enzyme to make it into DNA. That's reverse transcription. So that's what a retrovirus does. Other RNA viruses just infect the host cell, and they kind of start from here, where they serve as the messenger RNA that makes a protein. Or some work as RNA that can make another RNA that then makes the protein. But the viruses that are retroviruses start with RNA that then gets converted into DNA, that then gets converted into, that's supposed to be an M, messenger RNA, that then gets translated into a protein. So here's the viral RNA. It will actually use reverse transcriptase to make its to make the DNA, and then I think it uses the same enzyme to make the double-stranded DNA. And then that goes into your nucleus and inserts itself in there. Not super awesome. Anyway, there it is, sort of just like the lysogenic cycle for bacteria. And then this DNA just provides it just works like regular DNA while it's sitting in your chromosome there. And it just provides the instructions to make messenger RNA. And so here's that messenger RNA. And it tells us, it uses a ribosome to do translation to make proteins. And here are the proteins. And some of them actually will just get packaged in those proteins and then leave the cell. So that is a retrovirus. So retroviruses use reverse transcriptase, and that is part of what's in the virus. That's going to be packaged in here. So one of the proteins made is going to be reverse transcriptase here. It's this one. So this is reverse transcriptase, the green one, and the others are like the coat that goes around the virus, the protein coat. All right, I kind of said this. Here's reverse transcriptase. Has like a part that works as a polymerase. So yeah, so anyway, this is showing you having the virus integrated into the host. So here, the purple thing with the orangey, I don't know, salmon color or whatever, this is the host DNA, and this is the virus DNA. And the virus DNA has some ends here, and it'll cut the host DNA and insert itself. So here's the virus, and it's inserted itself into the purple stuff, the pink stuff, the host DNA. So when it's integrated, it's not called a prophage, this because a phage is for bacteria, virus for bacteria. This is just called a provirus. So that's an R. A provirus is, I guess I should capitalize that too, huh? A provirus is a virus that's been inserted into the chromosome, into your DNA. And then here is the DNA producing RNA, just the normal way that we do that, just regular old transcription using your enzymes.